Filtration is the mechanical separation of solids and liquids from a suspension. There are many different types of filters that differ in pore size, structure, and material composition. In our laboratory, we most commonly use filters with a pore size of 0.45 micrometer, which are optimal for filtration of samples prior to instrumental analysis of major and trace elements. Use syringe filters to filter rather small amounts of sample, up to about 30 milliliters. Prepare a suitable sized syringe and attach the filter. Never touch the bottom tip of the filter to prevent contamination of your sample. Pour the sample into the syringe and insert the syringe plunger over the prepared clean tube. Filter the sample by applying adequate and continuous pressure to the plunger. It is important to keep the pressure constant and not to loosen the plunger, otherwise air will be sucked in and filtration will be much more difficult. The used syringe filter is disposable and should be removed to the waste bin. Recycle the syringes for further use. For larger sample volumes, use a vacuum filtration apparatus. The filter apparatus consists of a bottom collection vessel, a special lid, a top funnel reducer, a filter frit, and a top funnel. The individual parts are screwed together and a rubber seal is provided to each part. It is crucial to ensure that all parts are perfectly sealed to maintain the vacuum. Press the filter frit loosely onto the top reduction. Place a suitable filter on the frit. Remove the filter from the box. The producer usually supplies the filters in a way that each filter is separated from the others by blue paper, preventing multiple filters from sticking together. Do not rotate the filter, but leave it in the same side orientation as it was placed in the box. Now carefully screw on the top funnel. Make sure that the filter stays in position and is not screwed in during the screwing process. Connect the vacuum hand pump to the apparatus via a rubber hose and check whether a vacuum is generated. Pour the sample into the upper funnel and pump until the solution starts to flow into the bottom vessel. Use the pump to push out the last drop. Release the vacuum using the control lever on the pump. Unscrew the bottom vessel and transfer the sample into the appropriate sample tube. Unscrew the top and remove the filter. Discard the filter or use it for analysis of solid particles trapped on the filter. Disconnect the vacuum pump. The apparatus must be washed before filtering the next sample. Disassemble the apparatus into its individual parts and wash each part thoroughly and leave it dry in an oven at a maximum temperature of 60 degrees Celsius. After filtering the whole set of samples, wash the apparatus thoroughly and place it in an acid bath for 24 hours. Wash with distilled water and dry in the oven.